Okay, so standard reminders for this week, again, as with all weeks, the muddiest point due Sunday. There's also a concept check over topics that we're covering these first couple of lectures due this Sunday. This unit is a little longer than the others, so it's got a second concept check. It will be due in early February along with mini project one. Nice work on the muddiest points for last week. I hope it didn't cost you that many minutes of time. I'm just looking for a couple of words for question one, a couple of words for question two, and then a no or an NA for question three, or whatever you'd like to expand on. And I tried to do my best to respond to everybody individually. There were, I think, only like 3 submissions that were a little suspicious in that they commented on topics that we haven't talked about yet. So I had to deduct points on that. It's supposed to be like an all or nothing kind of completion thing, but as students are now using more and more AI browsers, then we start getting these nonsense responses that because the browser, it knows we're in unit 1, but it doesn't know what weeks they're in. So then it starts saying that the muddiest point for this week were things that we'll talk about on Thursday. So for those, I have to sort of start moving towards a zero tolerance policy. So if I start seeing submissions that just don't make any sense whatsoever why they be submitted, I'm just going to have to start giving zeros to them. But everybody else, it was clearly they sort of were relevant and voiced individual interests, confusion, whatever. And so I tried to respond as best I could to those individual questions and questions that came up more and more. I put that little note that I sent an announcement on. So there was a question about, you still be confused about the difference between heuristics, metaheuristics, and hyperheuristics, and how the use of heuristic in an optimization case relates to a more specialized use of heuristic, let's say in the AI or AI planning cases. So people brought up A star, for example. So if you've learned about A star, if you haven't, that's fine. But it's a way of finding these sort of shortest paths through graphs, and it makes use of this heuristic function that calculates a approximate cost to go. And if it happens to underestimate the real cost to go, then this is guaranteed to give you an optimal solution. So why is that a heuristic, given that that's guaranteed optimal? So I wrote up a little thing that provides that extra context. So if those are questions that you're having that I didn't personally address as well as you'd like in your response to your muddiest points, take a look at that first announcement, that latest announcement on Canvas that links to that little write-up that I put together, which goes through in a little more detail what the 3 different levels are, what the difference between parameters and hyperparameters are, and how it relates other uses of these terms you might have heard in some AI classes. So that's out there. Yeah, so I guess any questions about the deadlines or anything here? Administrative stuff. Yep.
How do you guys grade the mini projects? So you have to implement. I looked over something, and I know we have to implement algorithms based on certain criteria.
There's a rubric, and so the grader will go through and use the rubric to. So the mini projects, the first mini project is, I actually have a set of questions that's sort of a designed exercise. All the other mini projects, they're basically open-ended, where they're going to say, for this unit, sort of design a 1 to 2 page, design something you can report on in a relatively short frame, like a 1 to 2 pages or something. I think it gives an example that where you implement an algorithm from this unit and demonstrate something novel or interesting about that algorithm. And so the key there is there's some of these algorithms already implemented, and it says so you can use API for auxiliary functions, but I don't want you to just download like the genetic algorithm toolkit or something and just have it do the GA. So if you need tools to help you with some subcomponent of it, to calculate some sort of optimization function that you're using your whatever your algorithm is to optimize, that's fine. But making a sort of a simple version of the algorithm that shows that you understand how that algorithm works and what are maybe the limitations of that algorithm or the strengths of that algorithm. So it'll be a little bit more open-ended for each one of those mini projects as how do you want to do that demo? How do you want to sort of create that But, like you might want to say that, oh, this algorithm is really good at diversity preservation. So I'm going to show that for this population-based algorithm, I might have a baseline that I'll just use a stock genetic algorithm. Then I'll use this algorithm and I'll say that, oh, look, the population stays like really diverse in this algorithm, but not so diverse in this one. And then if you just like were able to sort of capture that in a plot and comment on it in an intelligent way, in a compelling way, then that would be a fine solution. Does that make sense?
Yeah.
Yep.
I have one more general question. Is pretty much every like assignment going forward going to be due Sunday night?
That generally speaking, I mean, there's probably some exceptions, but yeah, generally it's going to be rounded to wherever we reshare this. So it's going to be something like I'll look to see when the unit ended, and then I'll try to add enough days that I feel like you have enough time to complete the whatever's in the assignment, and then I'll round up to the next Sunday, and then I add the grace period of Monday morning. So that's usually the way I set those boundaries. But all tentative deadlines are all on the Canvas calendar right now. So you of course can submit them early. If Sunday's not working for you, and I try to provide enough buffer that I think you'll be able to do them by Friday or Saturday or whatever. But I thought usually make them do in the weekends. The end of the weekend I thought is better than the middle. Other questions? Share. Code. Go. Okay, so as last time we talked about the basic introduction to the idea of evolutionary optimization metaheuristics, and so we're going to see a little bit more about what we mean by these population-based methods and what we mean by evolutionary methods and what it means to have reproductive competition as we drill down into these concrete details that we're starting to open up now. So today, we want to provide some of these fundamentals of population and quantitative genetics that apply to the genetic algorithm. So this starts to give us the vocabulary that we need in order to talk about the GA, the components of the GA, and why they're structured the way they are. And as we go through these are going to start out sort of more biological, but I want you to look at this content through the lens of an engineer or a computer scientist. So these will be things describing how we think life works. But as a computer scientist or as an engineer, you sort of think about like, well, what do we think? What function comes from that? Like, how does this work? Why is this working the way it does? What are the potential limitations here? Why does biology? When do biological contexts need to worry about those limitations, and when do they not? Trying to think about that in a more mechanical way. So that's just something that, as we go forward, that's the perspective I'm hoping you're getting here. All right. So the core idea that we're going to start with here is, that we're trying to build up what does it mean by evolution. So that's the context here. These living systems under evolution, and so we're going to use this term traits. Traits here, organisms achieve function. So function is useful stuff. So as engineers, somebody says, I need to be able to fly. So we say, okay, I need to build you a mechanism to fly, whether it be a jetpack or a pair of wings or whatever. So the different ways in which you can give people options to fly are traits, but the actual function is I fly. So there's different ways to fly. The different ways to fly are the traits, but what's actually happening is the function. So organisms achieve function by traits, and we use the term that they express. So traits are observable and measurable. So the idea behind a trait is that it is something that could provide function, but it does not necessarily provide function in the current context. Like it's just something that could be used. So as an example trait, brown eyes, I think I have brown eyes. We call that a trait because it's a specific state. But the group that fits into, like I could have blue eyes, I could have green eyes. That's the character. So the character is the thing you're measuring, and the trait is the measurement value. So the trait is the thing you actually observe. The class it fits into is the character. Traits can be categorical or quantitative. So I already mentioned that eye color. So that's an example of a categorical or a qualitative trait. So they come from a discrete set. So there's no metric scale. You can't say, for example, that brown eyes are farther away from blue eyes than green eyes or something like that. There's no distance between the trait values. And that's what makes what I mean by saying no metric scale. So traits can come from a discrete set and have no metric scale between them. They can be two states, on or off. Like, whether your earlobes are connected or not, that is a yes or no question. Whether you can roll your tongue or not, that is a yes or no question. They can be polytomous, or this is sort of, so because we have dichotomous, some people say polycotomous, but that it turns out that that's not like proper Greek or something. So polytomous or polytomous is something with more than two values. So 3 or more states. So that's like eye color. And then you can have them be ordinal. So in this case, distance doesn't matter between them, but there is an ordering. So this is often things like disease severity. So you might say that an organism is born, and you can measure whether it has maybe a certain developmental severity of certain developmental diseases, for example, or developmental disorders. And you might say low, medium, or high. So in that case, you can still group the organisms into these 3 different groups, and they have a natural order to them, but there's no metric distance between them. So that's what we mean by categorical or qualitative traits. Yeah.
Something like small, medium, large?
Yeah, small, medium, and large. So these are often, ordinal traits are often more subjective, and this is in more of a clinical setting. So a lot of times in medical genetics, medical informatics, then they talk a lot more about ordinal traits. When we're talking about life sciences, so more fundamental biology, it's hard to find things that are clearly ordinal, like whether your earlobes are connected, that has biological meaning. But a lot of times these ordinal ones are quantized kind of inventions of a measurement apparatus, usually in a clinical or medical setting. So rarely is it's hard for me to come up with an example off the top of my head of something where like small, medium, and large somehow leaves, it's a little lacking, because you could say, well, can I define by, like, if it's small, like, didn't you measure something to determine it's small? And so you could say like that, okay, well, so not, I could actually then say, well, okay, 5 inches, 6 inches, and 7 inches. So at that point, it does have a metric scale, and we wouldn't call it qualitative. So it's usually things like severity. So again, disease severity, like a doctor does a clinical assay and says, well, it's a mild case of X, or it is a severe case of X. And that's where that comes from there, because they can't really put a metric scale on it. That's where ordinal traits come from. So that's one type of trait. And so you can imagine in engineered systems, like these are things that you express. So these are like in a design, like how many, so the how many doesn't apply to this, that's kind of the next one, but we're thinking about like if you're designing a car, does it have 4 wheels or 3 wheels? Is it a blue car? Is it a red car? Does it have a glass windshield, a plastic windshield? These are all traits these may provide function to the design, but they don't, there would be another jump to sort of say that, oh, well, the plastic windshield is better than the glass windshield because we really care about the weight of the car, and the plastic windshield is a lot lighter. So that's the point where we jump from traits to function and fitness, which we'll get to. So that's one half of traits. The other type of trait we have are so-called quantitative traits. So quantitative traits or characters. So that's sort of the kind of frustrating thing about it, is that technically all of these should be like quantitative characters and qualitative characters, because the traits are the state values and the characters are the variables that they fit within. But it just is kind of, for historical reasons, people refer to them as traits. So they call them quantitative traits. And but you just have to remember that traits are the actual values, brown eyes, and characters are the category of eye color. So a quantitative trait is one that does vary over a metric space. So you can define a metric that puts distance between the trait values that are meaningful. So height is an example of that. So you can say not only that you are taller than me, but you're 3 inches taller than me, and someone else is 4 inches taller than me, and someone else might be 2 inches shorter than me. So that height, that there's a distance between these heights. Same thing with weight, and also voice frequency or pitch. So you can say that my voice, there's a fundamental frequency of my voice that is lower than yours or higher than yours, and so on. But you can actually measure the distance in hertz for that. So that's what I mean by a metric there. So these are often continuous traits that may take any values in an interval and into a continuous real interval, like height, weight, and so on, but not always. This could also be so-called heuristic traits. These are traits that come from a countable set. So they're, in some ways, they feel discrete, so they feel like they should be qualitative, but the distance between them matters. So low, medium, and high, the distance, all you can do is order them. You can't say that you're more medium or something like that, or you can't say what's the distance between low and medium. But you could say how many eggs does this species tend to have on average? And so, this species has 2 eggs on average in every clutch or whatever. This species got 4, this species 5, and so on. So you can measure that, and then you can end up talking about the lifetime reproductive success of the animal, where the quantity actually really matters. So, that's, or like in mammals, the key feature of mammals is they all make milk. So that's something that's in common with mammals. But there are traits that differ, like how many nipples do mammals have? Like a dog, it has, sometimes like, especially for male dogs, like people think, oh, what are these weird little spots on the bottom of my dog? And, they're funny moles or whatever. And then they go to a vet and realize, oh, those are its nipples. And there's lots of them. So, that's an example of a countable trait that can vary, and it varies in a potentially meaningful way, which have to do with litter size and stuff like that. So this would be like the number of wheels on a car. Does it have 2 wheels? Is it unicycle? Does it have 4 wheels? Is it a massive truck? The number of wheels could be potentially meaningful, and you can not only order them, there's actually a distance between them. All right, so any questions about those 2 categories, qualitative and quantitative traits? So at the beginning, early in genetics, everyone was focused on qualitative traits. If you learned about Mendel's laws, the pea pods or the pea plants and so on, then it was all, are you, this color or that color? Do you have this many leaves or that many leaves? And it was that sort of thing. Do you have it or not? But then that became confusing, especially to those who were sort of, in Darwin's circle, because most traits that you observed are these traits. They are these quantitative, continuous traits. And that's what we're about to get to, genes, is that the mechanisms of inheritance all seem to be discrete, but most traits you observe all seem to be continuous. And for years, that created a lot of confusion until people got into what is known, which we'll talk about here in a second, as quantitative genetics. And quantitative genetics is how you get continuous traits from discrete genes. And so that's what we're about to open up here. All right, so questions about these two categories? Yep.
So is metaheristic always just a whole number and continuous can be any?
That's right. So countable. So yeah, it's having not going to be negative. So the counting numbers, the whole numbers. So yeah, like number of flower petals, that would be that. So but if it comes from an interval, a real interval, then it's continuous. Key thing here is in both cases, the distance between the trait values is meaningful, as opposed to just an ordering. Yep.
Can it be like multi-dimensional, or is it like one, like if I want to say something, like pink is X distance from red versus from orange?
Well, I would say that if you just said like the color of the eyes, if you just said pink, red, like if you just said there are like 6 colors of the eyes, that I'd call a qualitative trait. Now, if I said, what is the wavelength of the eye color? Then I've said, well, now I'm talking about a more refined description of the trait value, and it might be in that case, the wavelength of the color would fit into a continuous trait. So, there might be some traits that due to and this kind of goes back to I said like for clinical, like these disease severity, like it might be that we just don't have really a good way to measure that yet. So initially the best we can do is put an ordering on it. So it might exist as a categorical trait, but then as we get more and more technology, we find out that there's some biological precursor of that disease that, if you do a complete blood chemistry, a CBC on it, you get some metabolite or analyte out of your blood, then you find out that, it turns out everybody with low severity of this disease has white blood cell counts that are within this range. So then now that fine number, the white blood cell count, that becomes something that we can talk about here. So, and most of these, like, in some ways, those types of like fuzzy things that are kind of on the boundary here, it doesn't matter so much that they're there. The reason we have these categories goes back to that debate that I was talking about, like sort of on the bubble going from Mendel to Darwin, where they were trying to understand, everything they knew about kind of inheritance and trait expression up until that point was based on you have it or you don't. And then suddenly they're saying, but all of this stuff that we observe, most of the variation in Darwin, natural selection, we'll talk about all this stuff as it becomes important, variation was important. And all the variants you could observe in nature seem to be, or at least most of it, continuous. And so that's where they said, these things we can't explain with all of the legacy knowledge we have. So we have to bin them into another group. And that's where we got quantitative genetics. Nowadays, you can argue that almost everything is quantitative genetics. Quantitative trait locus, gene-wide association studies. If you've done any bioinformatics or will do any bioinformatics in the future, you're going to hear these terms. Almost every trait has multiple genes that are involved in it. And so this has become the dominant framework. But we still kind of teach basic genetics thinking about these types of traits. So there's sort of a historical accident that we have these categories. And if you find that there's something that's on the bubble, don't worry too much about it. Think about what's on the extremes. That's the reason why we have these categories. Question in the back? Okay. All right. So let's get more into the meat of it. I talked about a single trait. Now you aren't just a single trait. You're not just your eye color. A vehicle is not just whether it has a plastic windshield, or whether it has 4 tires. It's the combination of all those things together. And that's what we refer to as the phenotype. So the phenotype is the set of all traits. It's not the set of all characters, it's the set of all traits. Your specific eye color, your specific height, and so on. And then I just said height. So we know my weight, for example, my weight is not entirely determined by my genes, it's determined by what I eat, all sorts of other things. And so when we talk about phenotype, there are two components of phenotype. There's the heritable components, the genes, and then there's the environmental component. And so we refer to this as G by E to P. So this is when we talk about gene expression, or the expression of a phenotype. We're saying that it is the combination of two things. It's the combination of inherited components that mix with the environment, and this environment could be your current environment, it could be your past environment, and how it has left a plastic deformation on yourself. So you might have had an early child environment which changed your developmental history, which changes how your genes respond to your current environment. So all of that stuff that's not inherited, we've been in this E, and then we have the G over here. So a big part of understanding trait expression is factoring these two out. So there's all sorts of different methods that biologists use to try to figure out how much of a phenotypic variation is due to gene variation versus environmental variation. And so that's where the term heritability comes from. You look at a population, you look at how much variation there is in that population's phenotypes, how much variation that is in the population's genotype. You correlate the two of them together, and whatever falls off the regression line you get is environment. Whatever is consistent with that regression line, the variance explained by that regression line is the genetic component. So G by E to P, you'll hear people refer to that. So like some people say, oh, but I have the gene for this. And you say, and so they might say, well, what do you mean you express that trait? Because and that's why we don't usually want to refer to I have the gene for. You like, let's say for example, that blue eyes required a certain pigment from the environment and required you to have a gene that uses that pigment to express blue eyes. Well, what happens if you can't find that pigment in the environment? You move into an environment where that pigment just does not exist. Well, you have the gene for blue eyes, but your eye color will not be blue. So, and there's a bunch of examples of this sort of thing here. So G by E is really important. And like I was saying, you can have the same G and a different E gives you a different P is what I'm saying here. And, in this, I think, example I did with hair color. So if your hair color requires a pigment from the environment, that pigment's not there, your hair will express a different color. So, that's, if I want to sort of think of an engineering example of this, so I grew up in a day where computer games were all software timed. And so if you played a video game in the 80s, for example, the code that was running that video game would wait, like it would sleep for a certain fraction of a second in order to keep the timing right. Or it wouldn't necessarily sleep for a fraction of a second. They might have a little for loop and it would go like through 100 iterations, and that would be the delay. And that worked really well until you tried to install that software on faster machines. And then it became completely unplayable. So that was an example where the code, that's like the inherited component, that definitely was important in the phenotype being how fast, the playability of the game. But the environment, that code had to run on different computers, and if I ran it on a computer, that for loop was executing faster, and the delays were all off. So that was an example where you really, in order to get the correct phenotype, playable game, you had to play it on the old hardware. Then you got hardware timed games, and that started where you actually would sleep for a certain amount of time, and that would fix that. Now, interesting case is that the same, there was this, these games that I can't think of it now, the game where you fire up at the asteroids coming down, maybe asteroids. What was that? Galactica, yeah. So you've seen, old, Atari games or arcade games where you've got these asteroids that are coming down to you, and you've got the little spaceship in the bottom, and you're moving down to the ground floor, and you're firing up at them. An interesting case of that is that initially the way they built that game, it was running on really slow hardware, and so it would have to draw all of these space invaders or asteroids or whatever's coming down at you, and it would take a while to draw all of those before it would allow you to move your little spaceship to fire up at it. So you actually had a lot of reaction time available to you initially in the game. But then as you started to blow these things out of the sky, so there are fewer of them, then it had less to redraw. And then now you had less time to react, and it made the game harder. That was not something the developers built in. It just naturally emerged from the software timing of that loop. And people liked that because it got it kind of harder. When they upgraded that onto new hardware where it could draw these things rapidly, then you got the same reaction time regardless of how many things were being drawn on the screen. And people didn't like that. So they had to then add that into the code because they lost what they got from the environment. So that's another example of how this applies to biology. You get some things for free from the environment. You get certain dynamics for free, you get certain compounds for free, and so on. And in engineered systems, depending on what you're building them into, there are certain aspects of the observable features of your engineered system that you get from the environment. So how fast the hardware runs, how bright the projector is, and so on and so forth. And if you then start shifting to a different hardware platform, if you want the same phenotype, the same trait, the same observable metric of your design, you may need to change the genes, change the code in order to make that work. So that's a way that this analogy this works there. Yeah, good question.
Does a character have just one phenotype, or does it have multiple phenotypes?
Well, so a character is so I would say that, so it's kind of like your phenotype, there is a vector, your phenotype is a vector. Is a vector. And that, and each box has got traits in it. So this is trait 1, trait 2, and so on. So this is a set of traits. Now, each dimension, so these are all of the possible traits for that dimension. So these are other possible traits for this dimension of the vector, for this element, the variable of the vector. This dimension here, this variable, that is the character. So the character is, the phenotype is a set of traits. Each individual trait is a value of a character. So if you think about all of the possible traits that a species can express, then you're sort of getting into like, what's the set of all the characters? So if, so I don't know, there is no good name for the set of all characters. But if you were to sort of say like, well, what does it mean to be a mammal? Well, then you can say, well, there's certain things I observe about a mammal, certain things that are in common and certain things that can vary. And all of the things that I can observe about a mammal, those are all the characters that that are useful to talk about when I talk about mammals. But each individual character is something like, how many nipples does it have? How hairy is it? Those sorts of things. Those are individual characters, but the particular value, so, 2 nipples, only has hair on its head, etc., those are traits. So we don't think of character as somehow, smaller. It's just a different category. So phenotype is a set of traits. Character is a dimension of ways in which an organism can differ.
So if you're talking about the human example, let's take human example of phenotype.
Mm-hmm.
You can have traits like hair color, eye color, and maybe skin.
Those would be characters. You can have characters like hair color, eye color. Individual hair color like black hair, brown eyes. Those would be traits.
Like, is it like values of the traits?
Values of the characters are the traits. So, so the character is the big set. And so character, let's say trait, it's like trait is an element of character. Well, I don't want to say character. It's like the character defines the set of trait values. So this is like eye color here, and this is like brown eyes here. Yep.
Would that be then the genotype? And when it gets expressed, that becomes the phenotype?
We're about to get into genotype. There is not a one-to-one genotype-phenotype mapping. So I want to be very careful about that. There is no gene for brown. There very rarely is a gene for brown eyes. There usually are multiple genes that maybe affect the expression of a particular trait. But some of those genes are involved in other traits, and so on and so forth. So we'll get to that in just a second. Any question about this distinction between trait and character? Sometimes character, you can think of it as characteristic, just for like historical reasons, it's called a character. You can think of, what's the characteristic? Well, the characteristic I'm looking for is eye color. Well, what types of eye colors are there? Well, there's brown eyes, there's red eyes, there's green eyes, and so on. So the values of the characteristic are the traits. The characteristic itself is the dimension of measurability. Okay, I did everything there. All right, so yeah, so now we have to get to genes. So we have this, this idea of the heritable component of a phenotype. So those are the genes. So there's some aspects of your phenotype you get from your biological mom and dad, and there are other aspects of your phenotype you get just through historical contingency, just your life and how it's accumulated over time. So we refer to genome this is another sort of hierarchy here. So we've got this genome, which are how the genes of a species are organized into so-called chromosomes. And so the genome are not the genes of a species, but they're sort of how the genes are organized. And those genes here, maybe I should move upwards here. So maybe if I start with the gene is, I don't know, really it sounds like I need to start with a chromosome. But the human genome is divided into these 23 chromosomes. So the genome is like a library, and that library has 23 books in it. And each one of those books are chromosomes. For our purposes of the genetic algorithm, it will rarely be useful to have more than one chromosome. So when we talk about the GA, these two things become kind of the same because we just put all genes in one chromosome. But the way biology works is across different species, they have different numbers of chromosomes. So their library has different numbers of books in it. Each book has locations in the book, and those are like pages in the book. And that's what a gene is, or a locus. So it's a particular location or a locus within the chromosome is the gene. And it's just an address. So when you talk about, someone's genes, you're usually talking about things that are located at that address. And so we'll get to that. This is that idea of genotype. So for right now, this is just an addressing scheme. So you're sort of saying what neighborhood you are in, that's like what species you're in. So we're in the human species, so that gives us a map. And how is that map organized? It's organized into, I guess maybe I could say like, we're in the city of humans, and that city of humans is organized into neighborhoods of chromosomes. And then those neighborhoods have address numbers, and those are the genes. So in order to understand, where a gene is, you've got to know what species you're talking about, what chromosome it's on, and where it is in the chromosome. And that's how we get down to addressing into genes. It's just a location. And these, these things are then the things that are actually inherited and reshuffled and so on. So this is the stuff inside you versus the environment, which is outside you. All right, now, those genes, you have different things that could be stored there. So at a particular location, you could build a house, You could tear that house down, put up another house. In a library, you could go to, I could go to bookshelf 6, and from bookshelf 6, I could grab book 46, and on book 46, I can look at page 23. Okay, all of that's fine, but what's on that page 23 can vary. And so the specific information that is stored at the gene location, that's a so-called allele. It's from the Greek, I think, for other. It looks like allo, other. And so the idea here is that you can have multiple alleles. I can have an allele, you can have another allele. So it's the idea that at that particular location, so if you go to my chromosome 16 and go to a particular gene on my chromosome 16, it will have a certain bit of information at that location. Your chromosome 16, you go to that location, may have different information at that location, different things written in the book. And those are the alleles, the specific gene variants that can differ across the population. All right, now, those alleles are themselves made of strings of nucleotides. So if you've not taken, so this is something that comes up, is that there's some confusion about, you hear about these nucleotides, ATGC, and there's, I think, sometimes confusion about the difference between a nucleotide and an allele. And so the alleles are not the individual nucleotides. The nucleotides are just letters in the book, and the allele is like the page of a book. And so it turns out if you go to a particular location in a gene, there's a molecule, there is a protein that reads the chromosome, and it will go to that location, and then it will look for a sequence of nucleotides that are universally understood to mean start. And it will read until another string of nucleotides that represent stop. So these are tokens on that page. So that means that the amount of text you can have on the page can vary between me and you. So you go to page 26 and my chromosome 16, and I might have an allele that is this long, but then your allele might be this long. So you can go to these locations, and the size of the house at the location can vary. The size of the allele at the location can vary because of this idea of they're written with letters, and it's just like now, if you encode a string of text going into a neural network, you often have to have a token that tells the neural network when the text starts and when the text stops. That's the exact same thing that goes on in genes. So genes are written out of these letters, and then you can write different alleles. The alleles, the thing that they have in common is that they all have a start and a stop, and that allows you to write different programming code at these different locations. So these are just modules. This is just firmware that can be downloaded to different locations in memory, and that firmware can vary in size because of this start and stop. Does that make sense? So the nucleotides are not the allele. So it's not like I have the A allele and you have the T allele. That's not it. I have I might have something that is 1,000 nucleotides long, 1,000 base pairs long, and you might have one that's 900. But they're all located at the same location. All right, and evolution, this term evolution, is the change in the frequency distribution of these alleles. So at a particular gene, I can look across all humans, and I can say, at this location, page 16 on chromosome, which is like book 23 or whatever, I can then say, how many different pages do we have? And it may be that there's only 2. It might be that I either have allele 1 or allele 2. And there might be some of us who have allele 1 and some of us have allele 2 for that particular gene. And the question then is, what's the distribution of that? It might be that 90% of humans have allele 1 and only 10% have allele 2. Evolution is the change in that frequency distribution. So when we say that populations are evolving in a biological sense, what we're saying is that from generation to generation, we're seeing a change in the distribution of alleles in that. So if you were to say this particular set of alleles on this gene, we look at a histogram. So we've got all the 6 different alleles that are possible out there. And then that is going to form a shape of that histogram. If that histogram is changing over time, so one allele is getting taller, the other allele is getting shorter, then we're seeing evolution happen. It's not saying anything about whether things are getting better or worse, whether the better allele is getting more frequent or less frequent. We're not saying anything about that. All evolution is just that they're changing. And so the question is going to be, what causes them to change? And there will be certain things that cause them to change because certain alleles have an advantage, and there will be other things that cause them to change for other reasons. And so if we're designing an optimization metaheuristic that is based on optimizing function, then we need to be careful that we're implementing the thing that actually selects for good function. Because not everything in biology that is driving evolution is optimizing for function. Other things are happening. And we need to be aware of those things if we're mimicking biological evolution in our optimization metaheuristics. Yeah.
Could you say for evolution, the loss function is basically survival of the fittest? If you live, that's the like metric that you're going on.
I would flip it around. So we'll get to that. So the question was about how do we map this idea of a loss function? So in an optimization model, you might have a loss function and you're trying to minimize the loss function, bring it to 0. And is evolution trying to bring something to 0? I think it's actually going to be easier to flip it the other way around and think about a loss function in terms of evolutionary fitness. So in some ways, we were trying to use fitness to drive loss functions to 0. But it's more of we're trying to use, I don't want to say evolution can be conceptualized as a machine learning process. It's more the other way around. I want to say we can take a machine learning process and couch it as something that could be evolved. Like if we did happen to build a natural system, we could have that natural system evolve toward a good state. So it's more that I want the evolutionary thing to be the bigger set and the machine learning to be the smaller set. So we'll see that. Any other questions about this terminology so far? All right, so now we get to this genotype, a term that was brought up earlier. So the genotype is the complete collection of all alleles in an individual. I have a genotype, you have a genotype. It's relatively cheap for us to get our genotype now. Didn't used to be that, but now you can send in and you can get genotype. So that someone can say that we know that there's these books in the human library that have these pages, and being genotyped says we're going to read all of the pages of your library. And they'll say, at page 16 in book 22, you have this text, and I have this other text. And so being genotyped is reading out all those pages and seeing which pages you have versus which pages I have. So that's our complete collection there. So there is a function basically, and I'll just map this way, where I guess the inner function here, which I called a function pi, pi because it sounds like phenotype. So you can think of it as you have a vector, your genotype. Those are all of your alleles. And there is a set of interesting environmental things. It's another vector. It represents the current environment, like the current temperatures, the current, what you're breathing in, and so on and so forth. It represents your developmental environment, what happened to you as a child, what happened to you in the womb, and so on. That's a complex vector, and this is a complex vector. Those two vectors go into this pi, which is a function which produces another vector, your phenotype. So that's all of your traits. So this is the so-called genotype to phenotype mapping. And there is no one-to-one mapping between a genotype and a phenotype. Your genotype may be a vector of a very different size than your phenotype. There, you learn about very special examples, certain traits, certain characters, where we have identified one gene or whose allele variation directly predicts the trait variation. So there is one gene for the connectedness of your ears, there is one gene that has two alleles. And if you have one allele, your ear is connected to your head, your earlobe. If you have the other allele, it's not. There are a small set of those. Your earwax, whether it's kind of sticky or flaky, that's another one. Those are very rare exceptions. Generally speaking, you cannot find for a particular trait, there is no such thing as a gene for that trait. There is a pattern of genes that when you have that pattern of genes combined with the right environment, you get that trait. There is no gene for Riz, for example. You've either got there's multiple genes that correspond to the amount of Riz someone has. And it probably has to do not just with their genes, but with, what they learned from their older siblings, the experiences they had, and so on and so forth.
Are you sure about that?
Maybe for you. But I think generally speaking, you have to think of these as being decoupled and linked through this very complex function, which I'm arbitrarily calling pi. Figuring out what that pi function is a very active area of research. Now, once you have your phenotype, then the question is, what is its fitness? And so that's what this big F function is here, and that's this fitness, and that's the scalar. In biological senses, a fitness is the number of offspring that genotype produces on average. So I mentioned that phenotype is a product of environment. If we're assuming that everybody's environments kind of average out, then we can say for a particular, we can now look at particular alleles, and we can say through this mapping across the whole population, somebody with this allele might have a very different fitness than a different person with that same allele due to all the rest of this stuff. But we can say for a particular allele across the whole population, what's the average number of copies of that allele that we see in the next generation? And that helps us get this fitness number. So it is very complex how we get fitness of an allele, given that it involves all of the other alleles that are there and the environment that's there and the mapping from phenotype to fitness. And so I mentioned that, whether your car has a plastic window or a glass window, well, what type of, what is the function of that car? If it's an economy car, you're just trying to optimize gas mileage. You might want the lightest parts possible, and that, that provides you that value. If it is a car that is a sports car, maybe you like heavier components because they give you a lower center of gravity, which allow you to turn in different ways. And so this mapping of phenotype to fitness depends upon the environment as well. It depends upon what niche you're competing for. So we'll get to that when we talk about multi-objective optimization, this idea that in community ecology, you'll have different niches where in this niche, you're going to be optimized for a different set of things than in this niche. So here you might be optimized for fuel economy. Here you might be optimized for how fast you can go around a corner without flipping over. So those different things are going to affect how these alleles change their distribution over time. Questions about this idea? Yeah.
Explain the difference between the genotype vector and the character vector.
There is no character vector. It's like the genotype vector is drawn from, the question was, what's the difference between a genotype vector and a character vector? And it's almost like I have a particular genotype vector, I'll call it G vector, and it comes from a space of, I'll call it character vectors to the N here, so it's like, or imagine that all my characters could come from the reals. Well, then it would be like my genotype vector would be drawn from, so this would say that this would be communicating that the phenotype, or sorry, I'm sorry, you said genotype and the character vector. So, okay, yeah, or then I was thinking, again, I was going back to trait and character. So let's forget about this for a second. This.
Actually, I'm comparing them more to a schema of the organization of genes, so I don't understand the difference between them.
Yeah, so so like I said, the phenotype is an instance of the set of all possible characters. And the genotype is just the heritable components of the things that allow you to express phenotype.
So is it like a sample space?
I want to say that there's a bunch of different genotypes in populations, and when those genotypes combine with the environment, they produce different observables. And so this is like an internal latent state, the genotype here. So, and this could be gigantic, the dimension of this state. This could be very small, the dimension of this observable here. And so because of that, you can have lots of different combinations of different genotypes that produce the same phenotype, for example. So there's not this one-to-one mapping. Also, because of the involvement of the environment, it might be the other side of it. It might be that there's only one or two genotypes, but those different individuals have had different experiences, which cause them to have different phenotypes. So, you might have an identical twin brother, in my case, or sister, and you might have the same genes, but they might have, 40 pounds heavier than you. And that is due, now we would still say weight is a phenotype. So you differ in your phenotypes, but not due to your genes. Does that help at all? So it's complex, especially because when you first start learning about genes, you learn about these very simple examples like the ear-connected thing, where it's like a gene for a trait. And in most cases, there is no gene for a trait. Yeah.
So if there's going to be a new allele, is that like the nature's version of random mutation? It creates a new.
Yeah. That's the next thing we're getting into. So hold that thought. All right. So this is where we left off here. We got this fitness function here. And like I said, genes are fundamentally discrete. Each gene only has a finite number of alleles, but traits are often continuous. And this was a conundrum for a while until this field of quantitative genetics came up, which studies how combinations of genes and variation from the environment give rise to continuous quantitative traits. And so you can think of it as not just G by E, but more like G by G by G by E by E by E. So it's this idea that you might have a particular allele at one gene, but when it combines with alleles from other genes, then they express differently. So you might have there's this thing called epistasis, genes on top of their genes, epi on top of, stasis standing. So one gene is standing on top of the other one. So there might be a gene for baldness and there might be a gene for hair color. Now, you might have the hair color gene for red hair, and if you don't have the gene for baldness, the allele for baldness, you'll express red hair. But if you happen to have the gene for baldness, it doesn't matter what hair color gene you have because you don't express hair, you're just bald. So that's an example of once you combine these genes together, you get lots and lots more combinations of outcomes. Some genes mask other genes, some genes enhance them. And then you combine that and you have different environmental factors. So different individuals have different early environments, different individuals have different late environments, and so on. The early environments make deformations in you, they add fat, they change the way your brain is wired, and so on and so forth. And that changes how your genes react to later environments. So quantitative genetics is the field that studies how you get quantitative traits. And there's a lot of sophisticated statistics used in quantitative genetics. So if you end up doing any bioinformatics, you'll learn about things like QTL and GWAS. So these are methods which say for a particular trait, like whether someone has ALS, for example, then you can say which genes are involved and to what extent are they involved. And so you can look at massive data sets of people who either have the traits or don't have the traits. And you can look at genotype data across all of those individuals. And then you can generate these maps across the space of genes, basically across the human genome. And you can say at this location, variations in this gene are very predictive of whether someone has ALS. Variations in this gene are less predictive. Variations in this gene are somewhere in between. And you get these Manhattan plots. So they're like plots of a city. And those plots tell you that there are 15 genes that are involved in the expression of ALS. This one more than this one, and so on and so forth. And then from there, you can hopefully then come up with clinical targets and so on and so forth. So quantitative genetics provides us these statistical frameworks for us to understand how complex combinations of genes and the environment lead to these quantitative traits. Any questions about that? So this is the modern way we do genetics, quantitative genetics. Yep.
Will we be implementing like a GWAS in this class, or that's something?
No, that's not, this is sort of an aside, let's put it kind of in a box. But you can find libraries that do GWAS, and this is a very common tool set now. And I wanted to just put it out there so that if you see this language, QCLs, GWAS, that it will be like, oh yeah, that has something to do with quantitative genetics. And like I said, this has already been built. We're looking for sort of bio-inspired algorithms, and this is sort of algorithms for understanding biology. So it definitely intersects with machine learning. Yep.
Basically, it's the resultant set of phenotypes from G cross E that we're looking at in quantitative genetics.
In quantitative genetics, it's trying to understand how the genetic environment is producing the phenotypic environment. So understanding that mapping from G to P. So we can observe P, we can observe combinations of genes. So how do we then figure out that variations in this gene are driving more of this phenotype than variations in this gene? So that's what quantitative genetics is about, is looking at phenotypes and ultimately trying to understand what genes cause what traits. So you observe traits and then figure out the genetic basis of those traits. And if you can figure out the genetic basis of the traits, you can say, this particular cancer seems to be, those that are predisposed for this type of cancer have this particular set of alleles. So this fingerprint, so it would be nice if you could say, well, if you just have this allele, you're predisposed for this type of cancer. But in reality, it's probably that you have this suite of alleles. If you have that particular fingerprint, then you're going to be predisposed for cancer. So understanding what that complex combination of alleles is that predisposes you for these diseases then allows you to sort of say, well, what is that combination doing? And if you can understand what the combination is doing, then maybe you can come up with targets for therapy and prevention and so on. It also just helps understand, so there was a paper that just came out, I think in PNAS this week, that talked about this so-called, it's like an epistatic restoration of function. So it's this idea that you can have a gene that has had loss of function. So if you just look at the allele of that gene, it says that there's a part of you, a trait that should be broken. And if you look at another allele, it will say that based on the allele that you have there, there's another trait that should be broken. But if you have both of those loss of function mutations, both of those bad genes, then they found cases in yeast, and now it looks like possibly humans, that if you have those two loss of function mutations simultaneously, you actually restore both of those traits. So the idea that if you just look at one gene, it might look like you're broken. But if you say, well, if you've got this broken gene and this broken gene, together they work together, and you actually do have those functions. So that's what you have to kind of look across the genes in order to understand this gene to phenotype now. Questions about any of that? Okay. All right, so that gets us to the four forces of evolution, the things that we want to mimic or want to not mimic when we're building these algorithms. So evolution, again, is the change in the frequency distribution of alleles over time. The four forces of evolution, these come from population genetics, are mutation. This is the ultimate source of new variation in a population. So This could be, the random changes in DNA. This could be from cosmic rays that come out of the sky and they hit your genes, and if they hit, in particular, if they hit gametes, if they hit sex cells that can be passed on to the next generation, then they can have a lasting impact on, introducing a new allele that gets passed down. That tends to not be the main source of new genes, though. Turns out that all of the complicated copy, so they're just like a flash drive eventually dies over time, just like a hard drive, gets bad sectors that have to be marked and data be moved somewhere else. You can get copy errors that occur, and those copy errors that occur in your DNA are often the source of these mutations. So this is how we get, new across the world population. This is how you get new alleles through mutation. So we are going to need to mimic mutation in order to get innovation in our algorithm. Now, there's another way you get new variation, gene flow. So we're not going to worry about gene flow in this early unit, but as we start getting into distributed genetic algorithms, gene flow will become more important. So gene flow is this idea that you have kind of isolated populations, and one population has got a certain set of alleles in it. Another population, just because it's been isolated for so long, has a different set of alleles. Well, if something happens that causes individuals from one population to start finding the other population, then you can get a new introduction of alleles from those two populations. And that can cause a change in the distribution of alleles in both populations. So gene flow is the other force. So both of these add diversity to the population. So when we're thinking about innovation in our algorithms, we should think about milking these two structures, building something that has one or both of these to add diversity. Now, the other two are going to reduce diversity. And so one of them is natural selection, differential fitness, reproductive success. Differential fitness changes the relative representation of alleles. So if one allele causes an individual to have more offspring that also had that allele, then very quickly that allele will become much more represented in the population than other alleles. That's so-called natural selection. And if the population size is limited, this can lead to a loss of alleles. So in our cases, we're typically in our population-based algorithms, we're only going to have 200 individuals running around. Well, if one of those individuals has 10 offspring and every other individual only have 2 offspring, then over time, gradually that one individual is going to have more and more offspring. But if we keep limiting the population, so if everybody gets to reproduce, so the whole population size goes from 200 to 300, and then we say, well, we're going to randomly grab 200 of you to go in the next population. If you keep doing that, then eventually it's the one that is reproducing the most is unlikely to get lost. But those that are reproducing the least will be more likely to get randomly excluded by that restriction back down to 200 individuals. So if you have a limited population, that can lead to a total loss of a allele. So a loss of a genotype, a loss of a coding fragment from your population. And then the other thing that we have to worry about, which is related to small population sizes, is genetic drift. So these are chance random events or bad luck that happens to affect one allele more than another despite similar fitness. And so this is significant for small populations. And this is the idea that if a meteor comes out of the sky right now and it's going to hit somewhere, your genetic background does not matter. So it's one thing to say that, if I have superpowers, then my genetic background potentially gives me a great deal of fitness. And so that's natural selection. So then gradually, if I have superpowers and I reproduce, then over time, gradually, the next generations will have superpowers, and anybody without superpowers won't be around. So superpowers will take over. That's natural selection. But if a meteor hits and the superpowers have nothing, they're just as defenseless as everybody else, then it doesn't matter if I have superpowers or not. If the meteor hits me, it takes me out. If it hits somebody else, it takes them out. That's genetic drift. So there are random chance events. If you're walking through the forest, you're stepping on ants. It doesn't matter how special those ants are. Your foot is probably not differentially picking out certain ants, like only stepping on some and not others. It's probably just stepping on all of them randomly. That's genetic drift. And this is particularly an issue for small populations. Because like if you only had 5 individuals in a population, randomly, it might just be you're going to have mortality due to random chance that takes out one of those individuals. And that just happens every generation. You have roughly a population size of 5, and one of those individuals is just going to die by random chance. Well, that means you've got a 20% chance of random death, regardless of your fitness. That's genetic drift. And the interesting thing about genetic drift is that it can lead to fixation on a type that may have no fitness value. So let's imagine this case here. You've got 3 alleles, A, B, and C in a population, and they all have equal fitness. So they're all on average producing the same number of offspring. So one doesn't have any advantage over another. And it might just be like, it might be the color doesn't matter. So you have red cars, blue cars, and pink cars, and for whatever reason, nobody buys them any differently. So the color doesn't matter. So you just happen to have a mixture, an equal mixture of all 3 of those cars. Now, if a meteor hits, it's likely to kill the same number. Well, I guess the question I'm trying to ask here, but due to time, I'll jump to the answer. Is it likely to kill the same number of A, B, and C? And the answer here is no. The meteor is not going to respect the population demographics. So if it happens to knock out all of the A's, then it just kills all the A's and all of you're left with is B and C. So the meteor isn't going to try to balance itself so that it makes sure that it hits an equal number of A's, B's, and C's. It's probably by random chance going to knock out one of these more than the others. And so if that's the case, if a habitat can only, if that happens, or due to the restriction, if a habitat can only support 100, then would we expect A, B, and C to always be hit equally by famine? And if the population can only support 100, you might happen to get like, well, there might be 5 A's that die due to famine, 5 B's that die due to famine, but there might just happen to be 3 C's that die due to famine. It's going to be unlikely for exactly 5 to die of each one of these. And it's going to be small differences at first, but over time, it's going to cause one of these to be more represented than the others. So you might have that they all start out with 33, 33, and 34. But over time, it starts becoming, 30, 30, 40. And so over time, these differences, and as one becomes more represented than the others, then if you're throwing random darts at this population, it's going to be more likely that you're going to knock out all of the small population. And the large population, you'll knock out some of them, but they'll still have some members left over. So if you think about it, the stable populations of this Markov chain are everybody's A, everybody's B, or everybody's C. Because if a meteor hits and everybody is the A allele, then everybody's still the A allele. If you have a population limitation from 500 down to 100 and everybody's the B allele, everybody's still the B allele. So these are stable fixed points to this Markov chain. They're absorbing states of this Markov chain. And we refer to this in biology as fixation. So when a population converges to a single allele, typically due to drift. And so drift will always take over because natural selection will always equalize fitness over time if it's the only thing operating. And once everyone is of similar fitness, drift takes over. And it takes over in particular for small populations because they're the most sensitive to random events because they take out the biggest chunk of the population with the quantization, like when the number is small. Because if you take out one individual out of 1 million, it's nothing, it's noise. When you take out one individual out of 5, it's 20% of the population. So the smaller the population, the stronger the drift. And so this will cause, eventually you'll see, oh, my population's all my A allele. That must be the best one, right? It might be that the fitness were so similar, the natural selection effectively turned off and drift took over. And even though your whole population has one strategy, that one strategy actually isn't the better strategy. It's the one that just got lucky enough to get a little bit of a numbers advantage. And that numbers advantage then drove it to this absorbing state of this Markov chain, so that gradually the whole population was just that one. That's what we mean by drift. It's drifting towards these absorbing states, which biologists call fixation. All right. Any questions about that? Yep.
At what, how do you differentiate between drift and natural selection? Like, say if it was a war, I feel like there would be a mix of both.
Yes, and that's a great segue. Next time, we're going to talk about how you balance these, and that will get into, I think, the answer to your question. So I'll hold that to next time. I don't want to keep people too much. If there are any other clarifying questions, feel free to comment after. But otherwise, like next time, we're going to talk about the balance here. How do you build an algorithm based on this logic that is going to balance between drift and natural selection? And then gradually, we're going to start actually building that GA, seeing what the GA looks like as an algorithm. All right, thanks everybody. So see you next time again. Any other clarifying questions, feel free to come up.